I'd like to thank the conference organizers, particularly Andy Falter and Belinda Hansen, also Terry Gasterlin, for having us here to Prague. This was a rough trip to come here to Prague, torn away from our mundane duties back in the States, but here we are, and I hope that you guys are having a great time here. We certainly are. My name is Chris Cheadle. I'm the Director of Research for Biology Products Elsevier, and I'm here to talk to you about a product that we have which is called Pathway Studio. So, you know, what is Pathway Studio? That's a good question, and one that I hope that I can answer in this session and pique your interest in it. So you could say that Pathway Studio is a pathway analysis software. It certainly is that. You might you know, say, well, you know, it's really kind of like a molecular modeling software, and that's true too. And you might say, well, you know, it's just a great way to research the literature, and that's absolutely true. So it's all of those things, and it's a network-based tool. But, you know, when we think of networks, we're certainly thinking of nodes and edges, right? That's a network. And it's how you define the nodes and how you define the edges, which makes a particular network of a particular type. And we're really used to, in science, and thinking of networks as nodes connected by experimental data. So, for example, protein-protein interactions, things that have quantitation, or we could have correlation matrices, correlation measurements between, for example, gene expression. All of these things are based on data that has been experimentally derived. Pathway Studio, on the other hand, is basically like a first-order derivative, because what it is based upon is based upon the literature, okay? And so you might think, whoa, I'm not sure I want to get divorced from real data. But if you think about it, the literature is actually a summary of people that have basically done these experiments. And so, for example, in a protein interaction database, you might say A binds to B. In a literature database, you can say, you know, so-and-so says A binds to B. And guess what? 5 other people agree with him, okay? So that's essentially wraps up what kind of a database we have. It's a literature-based database, and it has a variable number of references for each one of the relations, and I'll get into the significance of that in a moment. We have a program that's been running now for about a year that's doing very well, and that's called Elsevier in the Classroom. I'll mention that again at the end of the talk. What we're doing is we're offering, this is particularly to instructors, we're offering to come in and provide free of charge the use of this software for the semester that you're teaching. We have no intent to take over the curriculum. This is not going to be a class about Pathway Studio, but we found in the work that we've done already is that the students love using this tool in the coursework that they're in the class that they're being taught, because it's a highly interactive teaching tool. We don't leave you alone. We don't bug you either, but we have a workbook that helps guide the students to, you know, become familiarized with the tool. We will come over and we will teach a class or two. We will find out what your subject is, and we will help design a little bit of a workflow just to get things rolling in your class area. And we've had a lot of real strong success in that. Right now we're at George Mason University. We started out in Georgetown. We're about to go to, of all places, Tianjin, China next month to kick it off. So, you know, it's an interesting course, and we can talk more about that offline at the booth. Okay, so before we get started, let's have a little bit of fun. We are here in the epicenter of the home of one of the greatest scientists of all time. Does anybody know who that is? Yes, thank you very much. Gregor Mendel. Gregor Mendel was, he's referred to as the father of modern genetics. He was an Augustinian friar and abbot in Brno, which I'm told is about 2.5 hours away from here because the roads aren't very good. They're basically redoing the roads. And of course, we all know that he basically came up with the basic rules of inheritance. So up until the time of Mendel, one of the big questions was, how do traits get inherited? And there was all kinds of ideas about this, and they were all wrong. And what really puzzled people, of course, was that traits seem to flash on and off in different generations. So if you want to put this in the context of how profound a contribution Mendel made was that in the absence of any biology that we would recognize from even a high school level today, okay, no cell biology, no biochemistry, no nothing, all right? He deduced what he figured had to be the rules of heredity, and he did it. It wasn't his day job. So if you all know the story, he basically was a beloved abbot of this monastery, and he taught school, like high school, in the town. And in the evenings, he went into a tent by candlelight, and he counted all those traits, all those beans, those peas, right? And he did this for years. And he realized that, you know, something was going on because the parental strains, the one father and the mother, basically were contributing what he called factors. They didn't even call them genes at that time. And he realized that basically it was the combination of these factors that was determining heredity. And then so in the famous case where they were recessive, that's how that kind of inheritance could be transmitted, not seen for a generation or two, and then suddenly show up as a double recessive, and you would see the trait. So not only, and he was absolutely right. No one has ever refuted Mendel. Mendel was absolutely right. He nailed it. Now, we also know that there's the caveat about whether these traits would have been linked if they were on different chromosomes, but the fact of the matter is he nailed the mechanism. And he also did it by basically counting. He counted the numbers and he deduced what the math must be, and he said, this is the way it must be. So, I'd like to compare and contrast Gregor Mendel with another great scientist of that era, was Charles Darwin. Charles Darwin was over in England about that time. We published a little bit earlier than Mendel by about 10 years, I think, 9 years, 56, and Mendel published in 65. And Darwin was a highly respected scientist, a well-known commodity, a member of the Royal Society, totally a giant in his own time, which he deserved, okay? And Gregor Mendel was an obscure scientist, and he only published one paper. And that paper was the paper that basically had the mechanics of heredity. And what, I'm sure you're familiar with those stories, but what you probably don't know is that when Darwin died, they took an inventory of his library. Because in those days, in the 19th century, if you were a gentleman scientist, which is what he was, you basically kept your own library at your house, and they went through all of his bound journals, and they found the journal that Mendel had published in, and on either side of the article, they found Darwin's notations in the articles on either side, and nothing in the Mendel article which suggested that he skipped right over it, didn't understand the significance of it. And for a brief moment in time, his fingers were actually on the pages that explained his theory of evolution. Because he had no mechanical explanation. His was basically a population theory. Mendel never published again. Was never considered an actual scientist. He had to have a real scientist sponsor him. And he was forgotten until the turn of the 20th century when people started looking at cell cytology, and they realized that chromosomes were segregating in a way that Mendel had suggested. And the rest, of course, is history. So, Gregor Mendel, certainly one of my heroes, and he lived right around the corner. And also, before we leave Mendel, he was not only the father of modern genetics, but I maintain that he was actually the first bioinformatician, because he did it by the numbers. He did it all by the numbers. And I think one thing that we have in common is that we're all, at some level, computational biologists, or if not frank, bioinformaticians. So, what is Pathway Studio? Well, we know that the scientific literature is increasing exponentially, over 1 million new citations in PubMed per year. So what do you do about this problem? Even an expert has trouble keeping up in their field, so one of the things that Elsevier has contributed to this solution is that we, using natural language processing, we do advanced text mining in a way that I will show you. How does it all work? So NLP is actually a bundle of algorithms which include dictionaries, okay? It includes recognition of sentence patterns, and it finally is a grammar. So the grammar is basically an integration into some kind of a knowledge base. So for example, if you were looking at this drug, pravastatin, which is a lipid control drug, look at all the different terms that you can find in the literature for pravastatin. Okay, so that's one of the tasks that Pathway Studio has, is that their NLP has extensive and dramatically enhanced dictionaries that will recognize almost any reasonable name for an entity. So when you get through this, basically you have to have a syntactic, so we're going from an unstructured text to a structured text, that's to a structured database. And so we have to do this by, the NLP, the basic unit of data is what we call a triplet. And the triplet is essentially a boiled down sentence with a subject, a verb, and an object. An entity acting on another entity is one way to think about it. And then finally, we have to integrate that information back into an understanding of a particular domain, and that's really where the art is. Okay, so triplet identification is absolutely essential, but at some point you have to integrate that into a knowledge base. And so what we've done, so where does it all come from? For us, it comes from everything in PubMed, plus we look at full-text journals, over 4 million, probably the largest database of its kind. We don't just reach into Elsevier, we reach into Elsevier and to other publishers for whom we have copyright license. And so let's stop there for a moment. So you asked me, Chris, do you cover all literature? No, we don't cover all literature. I calculate we cover about 2/3 of the major literature. And so the answer is that we're sampling, okay? We're sampling a tremendous sample. So you can all appreciate that as statisticians, that sampling techniques are useful when the problems are intractable. We get a very good sampling. So we may not get absolutely everything, but we get a good sense of what's going on. This has resulted in a knowledge base with over 7 million unique relations. It's big data. It's updated weekly, so it's basically timely. And so this is just the difference between information retrieval and extraction. Retrieval is what you get with a Google search, which is excellent, by the way, but it's essentially keywords and co-occurrences, okay? With a semantic analysis, you're actually getting information extraction. So just to give you an example of how the NLP works, you come across a sentence, say in an abstract that says, we have previously shown that S1 can activate GM-CSF and JERCAT T cells. And so what the NLP will do is that it will, let's see if I go for the advanced course here. Do I have a laser on this? I think it should be a bit more switched up. Excuse me for a moment. Okay, great. So it's this way. Okay. So what we have here is we have previously shown that S1. So basically the triplet will say, oh, this I recognize an activate statement. And we recognize that it's S1 and GM-CSF, which we know are genes and proteins, and we also have a directionality, okay? So one is going towards the other. So one of the big things that we do is we give you directionality, and if we can, we give you what we call polarity. In other words, is it an activation or an inhibition? If we can't call it, we won't. So we're kind of conservative at that level. So here's our network, okay? So we have entities, and we have relations. Entities like cells, diseases, proteins, small molecules, those are the nodes. And the edges are all the different types of relations that we have. It's an extensive view it's a generalist, but it's an extensive survey of the literature for those types of common biological potential relations. And we add new ones as time goes on, as the demand seems to appear for it. And I'll give you that an example of that in just a moment. So it's about networks and pathways. Here's a graphic look at some information. So the graphic look is very nice, and it allows you to look in a glance at a whole bunch of information, okay, and you get kind of a sense of what's going on. So for example, here is all the genes and proteins that are related to the disease schizophrenia that have a minimum of 10 references for the sake of making this a little bit more tractable. So that's a useful thing, and it gives you kind of a knowledge summary at that level. Of course, what we really want to do, and here's a legend for those different protein types, what we really want to do is we want to drill down, and most people like to use, serious players want to use the actual table. So this would be, analogous to the network table of nodes and edges. And each one of these has a relation, and you can drill down, you can see, for example, the most referenced gene right now with schizophrenia is CO-methyltransferase for whatever reason. If we can go look at another, perhaps a little bit of a smaller relation, we look at TNF. This also has to do with a genetic change. And we can drill down on any of these relations, and we can see what are the sentences that prompted this particular relation. So I don't know if you can read this, but it says, this study identified polymorphism in the TNF gene in a particular population in schizophrenia. And so that's the type of information that is used to build up that particular relation that says, you know that there's a genetic change in TNF that's related to schizophrenia, and there's 40 references that support that observation. If you get very interested and that's important to you, then you can drill down, and not only that, but you can drill down further, and you can actually go out to the PubMed or the actual full text document where that sentence came from. So, as I said, domains are, defining domains is really key. So, Pathway Studio started out as a frank gene expression microarray analysis platform. So we were obviously mammalian-centered, and it was all about genes. And then as time went on, we added in small molecules. We have over 100,000 small molecules, including drugs, antibody drugs, and metabolites. And we're going to go to 1 million next year from our Reaxis database. We've also added in a very interesting set of new relations. I've told you that we add in new relations and entities as we see fit and as we see it would be useful. And the drug development community really encouraged us to develop these relations for new biomarker discovery. Because, you know what, we have an entity called biomarker, okay, and, or a relation, excuse me, called biomarker, but that's easy. So a marker or a biomarker recognizes that right off the bat. But we have these three relations, quantitative change, genetic change, and state change, that would indicate that it might be a biomarker. In other words, because there's a specific measurable change in something related to that disease. And when we did that, we added in, of course, diseases, and we have over 16,000 of those. More recently, we have this cell effect module, which is in response to cancer immunotherapy. Very interested in immune cells and what's going on with them. So we added in the entity cell effects cells, and then we added in a specific relation called cell expression. So cells was not trivial. To give you an idea of what's going on behind the scenes. You know, for example, a T regulatory lymphocyte can be, it has a lot of names. And you gotta try to find all of them. And if you're an immunologist, of course, you want to identify them by the cell surface epitopes. But even there, there's a great deal of variety on how different scientists will use that to identify a Treg. So what our experts have done, they've manually gone ahead and basically standardized this. So virtually any reasonable term that you could use for a T regulatory site, it will be recognized and returned. So this is just an example of how useful this is. This is a disease called Preeclampsia. We can see that it's associated with these cells. The syncytiotrophoblasts particularly is the cell of the absolute lining of the embryonic villi, and it's the point of contact between the maternal circulation and the exterior of the placenta. And it's an interesting cell type because that's where the Zika virus, we'll be talking about the Zika virus today, that's where the Zika virus basically enters from the mother via macrophages that sit down on the syncytiotrophoblasts, and they are transported into the syncytiotrophoblasts and then into the embryo. So I already talked to you about the Elsevier in the classroom, so we'll just keep going. So here's a model workflow. So I want to give you an idea of how you can use this productively. And in this particular case, we were working with some collaborators on Zika virus infection and microcephaly. And as you're familiar with, the Zika virus basically causes microcephaly. So the question is, in neonates, so the question is, why would it cause microcephaly? Because there's certainly other kinds of encephalopathies, viral encephalopathies, that don't actually present in that, with that phenotype. So the question was, what could possibly be going on there? And so what we're going to do today is we're just going to identify genes that are upregulated in an experiment. We're going to explore the potential diseases that might be related to those genes. We're going to map major regulators. We're going to select a model target. And then we are going to map small molecule inhibitors of that target. and identify and distinguish between ones that are in clinical trials or are not. So first we import our data. So we have full import data function. What we're going to use is a data set from this available in Geo. And essentially what the researchers did is that they took neural progenitor stem cells and infected them with the Zika virus, with the idea being is, you know, what's happening particularly in neurons that would, you know, possibly, you know, lead to this particular phenotype. And then they harvested them at 56 hours and they compared them to the controls that were not infected. So we calculate our upregulated or differentially expressed genes. We basically have 476 of those, as it turns out. And what we then do is we do what's commonly done as a first step, and that is we just simply ask, you know, what is it about these group of genes that's interesting? So I'm sure you're all familiar with the ideas of enrichment analysis. Enrichment analysis, simply all you're doing is you're saying, against a standardized group of gene lists, you're saying in our gene list of interest, that is our upregulated genes here, is there any kind of enrichment for biological function or anything that would be more so than by chance? Okay, and there's some very, very basic statistics behind that keep track of that, Fishers or hypergeometric. So we have a wrinkle on this. We do the exact same thing, but we do it a little bit differently. We use something we call subnetwork enrichment analysis, which turns out to be pretty powerful. And what it's doing is that it's saying, you know what, if you're interested in diseases, we have a bunch of proteins, we have a bunch of diseases, and we have relations between those. And so what we'll do is we'll first go out into our knowledge base, and we'll basically make a subnetwork of all the proteins and all the diseases, okay? And they become the basis for our list, our enrichment list. And now we'll look at your proteins and say, is there any disease that's specifically overrepresented based on those proteins that we know about? So one of the advantages to that is that it's a completely unbiased approach. It looks directly to the literature. It doesn't rely on anybody's static gene list of what's involved in a particular disease. It captures all that information. And not only that, but since we update constantly, essentially it's an up-to-date search at any one time. And so in this case, when we do that, what we found was that we found, first of all, at the first few gene lists had to do with cancer, and that's not unexpected because, you know, so to a certain extent, when you activate these cells, they're hitting up a lot of different cell types. So cancer is often a kind of noise in these systems. But we also noticed that we had insulin resistance, and then we had, I can't read it here, but we wound up identifying 6 disorders of glucose metabolism, okay, all with high p-values. And we asked ourselves, well, that sounds like a pattern. So what's going on with glucose metabolism in these stem cells when they're infected with the Zika virus? So what we could do is for example, we could look at one of these. So this is the subset of our upregulated genes that were found to be related to insulin resistance. And you can drill down and look at any of this information. What you might want to do is essentially, we captured all 6 of these gene lists. And the first question we want to ask is, you know, how redundant are they? You know, because it could be like the same genes over and over again. Of course, to a certain extent, it might be, that's true. But it turns out that, in fact, when you ask that question, what is the union of those pathways, it turned out that there was 107 out of the 476 genes. So more than 20% of the genes, okay, of your upregulated genes has something to do with glucose metabolism. So we said, well, let's go down this path further. So then what we asked was, so that's the union, okay? And so what we wanted was the intersection. In other words, what is in every single pathway in our gene list? And what we found was that there were like, I don't know, maybe 8 or 9 of these. And by definition, these are major regulators because they remain connected to all of these diseases. And they're also in our gene list. And they include things like VEGF and insulin receptor substrate 2, all kind of interesting guys. So this is a lot of information to even this, which is a simplified network. And this is another feature that I really want to emphasize. We've really, you know, one thing with networks, I don't know if you ever heard of the term hairballs when referred to networks. I used to find networks to be very annoying because I could never figure out what was actionable in a network. You know, what's important? You know, they say, oh, that's a hub. Great, you know, and it was difficult for me to understand where I would go to from there. So we have invented a method which allows you to control the hairball, all right? And the way we do that is this is called our interactive network filter. And what it is it's really great. What it is it basically lines up everything that's in your network and allows it to be filtered all in one spot. So for example, we have two entities here, diseases and proteins, so we probably want to keep that. We have the number of literature references, okay, and I'll come back to that in a moment. And then we have all the different relations, and all of this stuff is selectable. So what we did for this look was, since we were interested in biomarkers, okay, potentially for this, we selected only one of these relations, which was quantitative change. If you recall, I mentioned that was one of the relations that we invented for or developed for biomarker discovery. And we basically said up here, the effect needs to be positive. So we could select for positive, negative, or no change at all, or no call. So what we've done is greatly simplified this network, as you can see. So while we're here and talking about references, I just had this conversation a few minutes ago. So the question is, you know, how reliable is the data in the network? And when we go around and around on this, you can do different types of literature metrics. You could, you know, scan, you know, who's a good author, who's not. But the problem with that is that there's no one-size-fits-all metric that would be very particularly convincing. Also, we like to be inclusive. You know, why would you accept a judgment on some? Again, we were having this. So some people say you should use impact factor. But you know, I don't know. I don't know if you've heard recently, like in the last 5 years, they've done studies, and the most reliable data is not in the high-impact journals. It's in the second level. Okay? So how would you figure that out? We make it very simple, and that is, it's a voting scheme. Just like what the algorithms behind Google are voting schemes, and this is a voting scheme too. And what we're simply saying is that if you have one person that says this, and it was 10 years ago, then it's, you know, buyer beware. If you have 10 people that said this independently, all right, well, you know, nobody's shouting them down. It's probably a pretty good observation. So somewhere in there lies truth. So the more references that support a relation, the more reliable it is in general. And we've tried many different literature metrics, and it always comes back to that, because that's just simply the most straightforward. Now, it is conceivable that the entire community is jumping off the cliff on this subject, and there's no way to stop that, okay? So you'd have to figure that out. But the NLP, does it always get it right? No, it does not, all right? It has about maybe a 10% false, positive rate, because sometimes it'll see a double, it won't recognize a double negative, for example. So when does that come into play? Well, you know, that makes single references tricky in some cases. But by the time you get to about 3 references, independent references, that kind of washes out NLP error, because it's unlikely that the NLP made an error 3 times in 3 different contexts on that subject. And so now you're getting into, so I usually, if I really want to just, you know, filter out, I'll just filter out to 3 usually. And also I'll go higher if it's a really complex network and I just want to know what's the fundamental observations in this field, okay? And that's really a useful way to do this. But we don't want to throw out single references. A majority of our database is actually single references. 2/3 are single references. And the reason we don't want to do that is because there's where your novelty is potentially. This is something that might have been published this year and nobody else has picked up on it yet. So, you know, why throw it out? We let the user control that. That's the bottom line. And so what happens is that when you do these complicated workflows, not that complicated, when you drill down, it becomes important to you if you're looking for actionable stuff. That's when you really start to get, you know, that's when you become more stringent on what you're accepting as truth. But in the meantime, you're using this as an aggregate tool, a filtering, just basically bringing in loads of information that you will then later sift and filter out. The filter is a great way of doing that. So behind this is, we basically identified, I should go back here a moment. So what we identified was that there was one, I don't know if I can find it here. Endothelin 1 is up here somewhere, and it remained connected to all 5 of those through this stringent filtering process. So we became interested in endothelin 1 as a potential, at the very least, a biomarker. Okay, and so And if we look at the tabular form, we'll see that, in fact, endothelin-1 is the most referenced gene or protein with diabetes mellitus and also with, I can't see too well here, but bottom line was that it was the most referenced for multiples of these glucose metabolic disorders. So we thought, well, you know what, endothelin-1, could be an interesting way to go here. So we focused on endothelin-1. So now, and this is basically working in our workspace, we have insulin resistance and endothelin-1. Now we know that there's a whole bunch of references that say that endothelin-1 goes up in response to insulin resistance. But the next question is, that's great, but just because it goes up, is there any information that would lead you to believe that intervention at that point would help to, treat insulin resistance? And so within the database, we basically looked for relations that went the other direction, in other words, from endothelin-1 to to insulin resistance. And we saw that, in fact, there was ample evidence, I think over 70 references, that said, yes, if you can push down endothelin-1 levels in animal models and in humans, you can actually reverse the course of insulin resistance. So not only do we have a good biomarker in endothelin-1, but we have ample evidence now to suggest that it could be a good therapeutic target of intervention. So what does the scientist do when they get to that point? They say, well, okay, endothelin-1 is a good protein target. You got any small molecules that inhibit endothelin-1? Well, yes, there's a bunch. So you can essentially, adding the network, you can find all the small molecules that have been discussed in the literature and that have anything to do with inhibiting, decreasing the expression or activity or abundance of endothelin-1, and there they are. In the meantime, you can also say, okay, for insulin resistance, are there any small molecules, or what are the small molecules that have been mentioned in any context in clinical trials right now for insulin resistance? So now we have two groups of small molecules, one that are endothelin-1 inhibitors, and those that have been mentioned in clinical trials. Now, clinical trials is a very useful relation for us. It goes directly, it's one of the few things that does not go to the literature. It actually goes to clinicaltrial.gov. And you can see that essentially we have this relation, and it'll pull up the clinical trial by clicking on that button in the relation table view. And in this case, we've chosen metformin. And you can see, I think you can see that here's the two diseases, insulin resistance and diabetes, and here's the drug. And you can read the record on this for metformin. You want to understand something more about metformin. Now, I chose metformin because it was it was the one that was most involved in clinical trials right now, and it's also an endothelin-1 inhibitor. So, if you want to know more about metformin, you can click through. We have links, extensive links to external databases, open source. So I really love this PubChem ID, compound ID database, because it gives you a really succinct description of the chemical entity, which is a biguanide in this case, and tells you what it's acting on, which means it winds up being a AMPK activator, and then it gives you a little bit about its clinical data. What's it being used to treat. Now, it turns out that metformin is actually a very interesting drug. So we'll take a little sidebar for a moment. And metformin turns out that it comes from Galega officinalis, which is a herb which grows wild. And it was used in the medieval age to treat frequent urination, which we now is associated with diabetes mellitus. So it was actually used in the Middle Ages as a drug to treat diabetes. And it was studied in the 20s, I think in the 1920s, and the active ingredient was identified as galegin. And you'll notice that it has this guanide entity over here, all right? And they went through a few evolutions. These were anti-malarials, and then they decided, you know what, maybe we'll make this into a biguanide because, for more potency. And they wound up developing in the 50s metformin, buformin, and fenformin. And they started using it for the treatment of diabetes. It was very effective. But then a funny thing happened on the way to the FDA. It turned out that these two forms of the biguanide were most potent, more potent than metformin in treating diabetes. But there was only one problem, and that was they were toxic at high doses, and it caused a condition called lactic acidosis. And so the FDA came in and said, you know what, we're banning them all. And they didn't bother to check out the PK data for metformin, which has shown that it's completely harmless. So they basically banned a very good way to go with diabetes and just said no more. So there's a happy conclusion to this, because in the 90s, they started repurposing. This is a repurpose of a repurpose story. They started repurposing metformin as an anti-neoplastic drug, and they used it in pancreatic cancer, because, you know, in cancer, they throw everything at the wall, right? And so they tried out metformin, and I don't know what it did for pancreatic cancer, but it cured diabetes in all the patients that had it, that had both. And they said, whoa, this is really good. So they investigated the PK data, and it was fine. They probably had to do that in order to get it passed as a tumor suppressor drug. And they said, well, you know what, maybe we should reconsider using this for diabetes, you know, seriously. Well, fast forward to 2012, metformin is the treatment of choice, first line for diabetes in the world, okay? So that's an interesting evolution of a drug, a repurpose of a repurpose. So it came right back down to, you know, they were right in the Middle Ages, and today it's used extensively. So, meanwhile, back at small molecules and clinical trials, we have those, and we also have the small molecule inhibitors of endothelin 1. And so what we can do very easily in Pathway Studio is we can simply overlay and extract and identify those molecules that are in clinical trials that are also endothelin inhibitors. Okay, and so I pulled those off to the right, you can see that. So, and what you can then do is you can reassociate the small molecules. So here are the small molecules that are in clinical trials for insulin resistance and also showing their relations as endothelin inhibitors. And this is figure level quality for publication. And so a lot of people like to use Pathway Studio for final figures. We call figure 7 because they're very pretty. And so if you think about it in terms of utility, however, you can see that we've pulled off over here. These are the ones that are already in clinical trials, and these are the ones that are in the literature for now. So in a sense, this is kind of a little bit of a drug development paradigm, because we've identified endothelin 1 as a good point of intervention, biomarker for insulin resistance. You can lower it, you can basically help to treat it, and we can show you, and this workflow can be done in a matter of minutes, we can show you the things that are already covered and the things that are not. Now, admittedly, this is a little bit of a simplistic, you know, workflow. But on the other hand, it shows you the complexity that you can get to in a hurry using this tool. And in fact, their major pharmaceutical firms are using this database behind their firewalls, okay, to do just this, just these questions. What are the potential targets? What are the potential small molecule inhibitors? Give us a range, because the idea here is that, you know, there may only be 1 or 2 actionable molecules at the end of the day, but they have to sift it down somehow to just get a list that they can inspect, things that they can look at. So it's very useful from that standpoint. And I think it would be useful for the regular academic researchers. So back to this question about whether or not, you know, does this hold water? So in other words, is microcephaly involved with disorders of glucose metabolism in the neural cells of these pre-newborns? And we do find some evidence, going all the way back to the 60s, this is from Betty Banker, who said that essentially everybody that she saw, okay, one of the effects of hypoglycemia in the newborn was in fact microcephaly. At the same time, in the New England Journal of Medicine, they also associated neonatal hypoglycemia with, among other things, microcephaly. More recently, they were studied in the 90s, metabolic control and risk of microcephaly among infants of diabetic mothers, and then perhaps even more convincingly, in OMIM, okay? Basically, they've associated genetic defects, certain genetic defects, with glucose metabolism disorders, and one of those phenotypic outcomes of that is microcephaly. So there is some, you know, stuff in the literature that suggests, you know, maybe something's going on there. But even so, I think that's a good example of how to develop hypotheses that can be tested out, all right? So, and this is just, you know, this is a cursory look. If you were a serious researcher, you'd have maybe half a dozen of these worked out and ready to, you know, to examine and test. Also, I think that, you know, when you're getting ready to do an experiment, and we're going to be doing this more in our development, it seems to me that the researchers of today, you never get into a project without examining the literature around your subject. Mainly because you don't want to basically be pursuing something that somebody already pursued, or, you know, come up with something wonderful and say, oh, by the way, 5 years ago, right? So you always examine the literature. So this, you know, basically examine the literature this way around your topic area. And then also, and I think we're going to get into this more in the coming years, you should and could examine the data. All right, so we have a new feature called Data Link, which basically allows you to search public databases for data by keywords. And we're looking to integrate that kind of a functionality into Pathway Studio because we think the two would go well together. You can search the literature, you can search the data, and you can integrate them right there in one platform in some kind of a meta-analysis approach. So that's what's coming down the road. That's what we're working on. So why Pathway Studio, right? So one of the questions we had on Friday was, I think from one of the conference organizers. She says, I'm going to ask a mean question. Of course, it wasn't a mean question. It was a perfectly good question. Is it why, you know, we know that text mining is getting important, really important. It's a hot topic. Why? Well, text mining is becoming important because there's too much to read. So, and we're already thinking AI, we're already thinking, you know, machine learning. So we're thinking like, you know, we got to get automated to a certain extent. And how do you do that? And he says, well, you know, there's reasonable solutions out there now, and they're free. They're open source. And I said, well, you know, that's a great question. So let's answer that. First of all, we've got scope. In other words, you saw the entire breadth of our relations. We go from promoter binding, expression. We have in our state change relation, for example, we can identify phosphorylation events between two proteins. We can, conversely, we can tell you whether or not an alternative splicing event was involved with a disease. In genetic change, we can tell you whether you could filter for a mutation, or an amplification, a deletion, or even an epigenetic methylation. And we have a wide range. So ours is a generalist tool. We're trying to capture broad aspects of scientific questions that people would be interested in from a biologist standpoint. And we have that. We have stability. So in other words, we're not going anywhere. Elsevier is a major multinational corporation and is committed to information analytics and specializing in health science and medicine. And this is our biology offering. And we are going to build this out, if anything. And that's a really important issue because stability is important when you're using tools like this because you don't want to be at the mercy of the postdocs finished up and gone. And also you can see that there's a tremendous amount of curation work, archiving work that goes on behind the scenes, the back end here. The back end is really well supported. Finally, we have content. We have a lot of content. We have PubMed, we have PMC, all of that. We have full text, and we're going to get more, and it's only going to grow. And we've already demonstrated that we're scalable. So in other words, we're just going up. Do we have errors? I've already said, of course we do. But we try to control those. We try to tweak the algorithms to get better and better. But to a certain extent, the error is the trade-off to having massive amounts of comprehensive information that you can actually, go through in an intelligent way. And then finally, costs. Our costs are going down. This is the good news for academia. It's true. Our costs are going down while other people's costs are going up. I will say no more. Because we are determined to make this available. We think, and that's why we have the classroom program, we think this is, and that's the feedback we're getting. This goes over terrific with graduate students. This is like a citation machine, if you think about it, on any subject that you can think of. It's a fantastic way to look at the literature, and we want to facilitate scientists getting a good grip on this stuff. These are complex areas. This is a lot. We don't do everything. We don't do it perfectly, but we do a lot. Okay, and we're committed to keeping our costs. They will be going actually further down because we're basically changing our business model to support A&G. So a lot of our business model has been into big business, and so that's a totally different cost structure. And we're now adapting to something that's going to be a little more realistic for researchers, of whom I count myself as one. I don't know if I mentioned to you, but I was core director of genomics core at Hopkins for 10 years prior to coming to Elsevier. So I want to thank you very much. We're finished right on time, time enough for questions.
Thanks. Are there any questions?
So do you have the ability to export, so let's say you have your network and you have the different papers that the interactions came from. Can you export those, like the papers from that?
Yeah, do you remember that relation table view that I showed you? Most scientists want the network look from the, yeah, that's totally exportable and selectable fields. We're also working, this year we're going to have a capability to export directly to Cytoscape, at least the network details.
Yeah, that was my other question.
Yeah, because and people keep asking me on the Elsevier say, well, why do you want to do that? And I said, well, we want to do that because scientists want to overlay different types of information. So you generate networks there and you pull them out over there.
So you'll have the ability to do XGML, do you know?
Well, we already have our own XML RNF formats, so I don't know exactly what we're going to do with the Cytoscape, but I think it's going to be, I forget the name of the file type, but it's going to be totally right into, it's going to be minimal information. So in other words, you're not going to get the full information. You're going to get the network information. In other words, you're going to get the nodes and you probably something like the number of references.
I guess the only other question is, how do you manage like users? So for instance, we have 800 users for software like this.
Yes.
How do you license it?
Okay, so we can do individually, or we can do site. I much prefer site, and I'll tell you why. I don't know if you're at a university or a college. Yeah, so we're really looking at site licenses, and I'll tell you why. Because, you know, if people get interested, and they do, why not make it available to everybody that has that email? It has privileges on that campus. And then nobody worries about it after that. And so when we have the student program, essentially the students, the feedback I got from students is they were shook up because they said, please extend our trials beyond the course because we have work we want to do now. And we did. Okay, we did do that. But the site license is by far the easiest. And the site license is very reasonable. It's not super expensive. So site licenses are the way to go. Individuals work for a while, I think, while you're persuading your institution. It winds up, interesting enough, going through the library. I don't know if you're familiar with that.
Yes.
Yeah.
We have two IPA licenses with Conway Library, and then our group has 2 licenses also, and it has to do with affiliate hospitals.
You know, I'm not sure about this. I don't want to get into it, but I've understood that some of those licenses are basically, you know, limited user. In other words, right? We wouldn't do that.
Yeah, you can use,
No.
so the way the library does it is you get an hour and a half once every 3 days.
No. Basically, we give unlimited use to all users, period. Anybody else? Great. I want to thank.
Thanks very much.
Thank you.